This is Lab 5, the fetal pig dissection, Part 3, Circulatory, Respiratory, and Nervous Systems, by Alyssa Geddis and Griffin Walker.
This lab was performed with the intention of gaining a better understanding of the relationship between the circulatory and respiratory systems of the fetal pig. Also, we collected data on the vital capacity of our lungs and the carbon dioxide concentration of our breath. This allowed for an analysis of the carbon dioxide concentration in the room air compared to exhaled air, both before and after exercise. The objectives cover what we will learn or identify during the fetal pig dissection, as well as what we will learn during the experiments of vital capacity and carbon dioxide concentration.
The respiratory system is responsible for the intake of oxygen from the environment. The circulatory system works in conjunction with the respiratory system to distribute this oxygen throughout the body. A buffer system is used in the blood to maintain a constant pH, using carbonic acid bicarbonate.
Our hypothesis is that the room air would contain the lowest amount of carbon dioxide, the exhaled air after rest would contain a higher amount, and the exhaled air after exercise would contain the highest amount of carbon dioxide.
The materials and methods. This lab was completed with no procedural changes according to the instructions provided in BB 2903 Lab 5, Fetal Pig Dissection Part 3, Circulatory, Respiratory, and Nervous Systems by Buckhold, 2010.
We examined the circulatory and respiratory systems of the fetal pig. Also, we collected lung capacity and carbon dioxide concentrations. Our results supported our hypothesis, showing exhaled air contained more carbon dioxide than room air, and that the exhaled air after exercise contained the most carbon dioxide.
This is a line graph showing the CO2 concentration of 3 different samples based on the number of drops of base that was used to return the phenolphthalein to pink. Your body uses more oxygen during vigorous exercise and therefore produces more carbon dioxide waste. This can be seen as the exercise air was consistently the highest concentration of CO2.
Table 1 contains the means and standard deviations for each lab section and the class's data for the vital capacity experiment. It shows pretty consistent means and standard deviations. The standard deviations were high due to averaging the male and female vital capacities, which were different.
This is our individual data. Alyssa was in the normal range at 3.6, and Griffin was above the normal range at 4.8. This difference was probably due to the averaging of the male and female vital capacities, which were fairly different. And these are our references. We referenced the lab and the dissection manual.